İkinci konuşmacımız Torino Üniversitesi'nden Enzo Medico olacak. Enzo Medico davetimizi bu toplantı için ilk kabul edenlerden, en hızlı cevap verenlerden bir tanesi. Ben birazcık onun özgeçmişiyle ilgili bazı şeyler söylemek istiyorum. Enzo Medico Torino Üniversitesi'nde tıp fakültesi okuduktan sonra La Sapienza Roma'ya gidiyor. Orada hücre ve gelişimsel biyoloji alanında da bir doktora yapıyor. Yani bir MD, PhD. Daha sonra çeşitli postdoc'lar yapıyor. Bunlardan bir tanesi Harvard Üniversitesi'nde genom transkripsiyon bilgilerinin onkogenomics olarak analizi ve kanser alanına, onkoloji alanına yansıtılmasıyla ilgili bir çalışma. Daha sonra 2004 yılında İtalya'ya dönüyor ve gene Torino Üniversitesi'nde onkogenomics alanında çalışıyor. Özellikle kolorektal kanserlerin onkolojisine yönelik çalışmaları var ve bu genom bilgilerini nasıl bir araya getirileceği üzerine. Aynı zamanda PDX, yani hastalardan alınmış kanser örneklerinin xenograft farede çoğaltılması, bankalanması, buna yönelik hem bilgilerin aktarılması, bu işlerin nasıl yapılacağına yönelik, akreditasyona yönelik kurgulanmış EuroPDX Research Infrastructure projesinin de Torino Üniversitesi'nde koordinatörü olarak çalışıyor. Ben sözü Enzo Medico'ya bırakmak istiyorum. Thank you, Enzo. The floor is yours.
Thank you very much. I wish to thank the organizer for the kind invitation, and it's really a pleasure to be here. I will share results and some reasoning about the application of genomic medicine and precision medicine on cancer. Okay, should work, should move. Yes. So let me start from the big problem. Many drugs that in development by the pharma do not make it to the clinics. And the reason of this attrition is mostly efficacy. So drugs that worked in the laboratories on cells, on animals, do not work in patients. And this has been the major problem. So what can we do to improve this and to improve our capacity of finding the right drug for the right patients? To improve preclinical models, so to make so that models more accurately reflect the tumor complexity and the tumor heterogeneity. And that's because we learned that the simple story of precision medicine, which is this one, you have a drug which binds to a target and blocks it. If the target is altered, then you can expect a response of the patients to that drug. And that is the basis of most molecular tumor boards and most precision oncology strategies. But this not always works. Actually, in many cases, it doesn't work unless it has been validated before. And that's because additional alterations occur in the genome of the cancer cells. Some desensitize, so renders the tumor resistant to the treatment, to that treatment. Others are sensitizing. And moreover, depending on the tissue, some drugs like HER2 antibodies, trastuzumab works on breast cancer with HER2 amplification, but it doesn't work on colon cancer with HER2 amplification. And there are additional elements of complexity that we need to put in the picture. We have intratumoral heterogeneity. So we take a small piece of tumor, we find it has a certain mutation, so it should be targeted by that drug. But maybe another area of the tumor doesn't have that mutation. So we need to take into account tumor heterogeneity and all the capacity of the intracellular signaling system to adapt to the drug, so that the cells the tumor shrinks, but the cell survives. We have a persister state where few cells remain, survive, and then finally give rise to relapse. I'm going to apply some example of this and how to improve all this workflow to colorectal cancer. So in colorectal cancer, you can see that depending on the stage, the 5-year survival is greatly variable. And I don't see the mouse, but it doesn't matter. You can see that in more advanced cases, the problem is that the treatments are not effective enough, and we need new treatments. In the early stage, the survival is very good, so many patients wouldn't actually need a treatment because the tumor is treated by surgery. Which cases need adjuvant and what adjuvant is needed. So these are the main clinical standpoints that we have to tackle and improve. How can we do this? Well, first of all, by using preclinical models that more closely recapitulate the biology and heterogeneity, in this case of colorectal cancer, and then by studying genotype response correlations on models, on many models. One way to do this has been, over the last 15 years, to generate patient-derived xenografts. That means you take a piece of tumor from the patient. Sorry, what's happening? It's not going back? Oops. Okay. You put it in, you grow it in mouse, immunocompromised mouse, so that it grows, and then you can study a number of things. You can study how it responds to different drugs. You cannot test multiple drugs in the same patient, but you can do that in the same tumor when it grows in the mouse. You can study tumor-stroma interaction. You can study metastatic evolution, can generate profiles and associate them with the response. You can also derive cells, organoids, and other things. So you study in vitro, in a quicker fashion, drug response. So this is a platform that, to be effective, requires many, many models from many different patients to recapitulate the heterogeneity of the disease. And to do this for many tumor types, we have been assembling. You see, like 10 years ago, we just celebrated the 10th anniversary, a consortium at the European level, which is called EuroPDX, that finally gave rise also to an infrastructure that provides the models or provides studies on the models on a collaborative basis. And this is the website, www.europdx.eu. We are many centers, and all together we assemble a collection of more than 2,000 models for different cancers. And we have more than 500 models just for colorectal cancer. So you can see here there is a snapshot of the different tumor types that have been collected, but this is all that this has to be, is going to be, is actually increased, but this is just to show you how we can generate models, multiple models for the same tumor and study drug response in many, many models. So clearly there are challenges. Not all tumors engraft grow in the mouse. Colorectal cancer, we were lucky, engrafts successfully, but other tumors like breast cancers and other do not engraft easily. And then there is the big problem, but when a tumor grows in a mouse, is it really representative of what happens in the patient? Does it maintain the features of the tumor when it grows in the patient, or something changes because it is in a mouse environment? So we had to also embark on this and validate the models and verify. So we embarked on a big project a few years ago in which we generated two cohorts, one of colorectal cancer, which engrafts easily, and one of breast cancer, which engrafts not so easily. And so you can see we had in total more than 120 samples for which we have the tumor of the patients, the patient-derived xenografts, the PDX, at an early passage, so just a couple of times after it had been grown and reimplanted, or at a later passage, so after the tumor had the opportunity to evolve in the mouse, okay, while growing in the mouse. So we have three cohorts, and we compared the genomic status of these cohorts at the level of copy number alterations. So colorectal cancer, in particular the non-hypermutated cancer, typically generates a lot of copy number changes, amplifications and deletions. And these are the GISTIC profiles. That means the statistical abundance of lesions at the different chromosomes. So you can see from chromosome 1 to the left to chromosome, so from chromosome 1 to the left to chromosome 22. And you can see amplification here and deletions here. And you can see the primary, the PDX early and the PDX late. And you can see that they are identical. So there is no change in the genomic assets of these populations of tumors over time. And this is good for colorectal cancer, but also for breast cancer. So which showed that these models maintains their genetic makeup while growing in the mouse, and that's very important, of course. So the copy number alteration profiles remain retentious. We found some cases where they change, okay? But we also saw that if we take two pieces of tumor and generate two samples, they can be different, and that's because you can generate the profile from one piece of the tumor and generate the PDX from another piece, and they are not identical because there is heterogeneity. And this heterogeneity is similar to what is observed in studies like TracerX, in which they have been comparing the molecular profile of different pieces of the same tumor, and they found heterogeneity, of course. So the conclusion is that these models are good. They are, in general, capable of recapitulating the genetic alterations of the human tumors and to maintain that in the mouse environment. And some examples, this is the response to cetuximab, a drug that targets epidermal growth factor receptors in colorectal cancer that do not have mutations in KRAS or BRAF, so mutation that would render the tumor resistant. So you can see we have some of these tumors that actually respond, so the tumor size is reduced by the treatment. Some others are stabilized, but others grow, and we don't know why. Okay, we didn't know. So the group of Livio Trusolino and Andrea Bertotti found that some of these actually had HER2 amplification, amplification over 2, which is frequently found in breast cancer, most of you should know. So, well, the basic idea is like precision oncology. It has HER2 amplification, so the mouse should respond to trastuzumab, to the antibody that is given to breast cancer patients. Well, actually, the mice did not respond. And that, unfortunately, also happens in patients. You find the lesion, you say, okay, let's use the drug, and the drug, unfortunately, doesn't work. But with the xenografts, we could understand why. And the reason, you can see trastuzumab and even pertuzumab given together, the tumor in the mouse just grows almost as much as the non-treated tumor. To actually obtain efficacy, you had to add to the blockade of HER2 the blockade of EGFR. Only when the combination is done, you see that the tumor shrinks. Okay? And in particular, lapatinib and trastuzumab proved effective. So only by using the same model with different drugs, you can understand exactly how to treat that particular case. But this is true just for this case, for this particular PDX, which is called 080, or is it good for in general? Well, they had a clinical study. A clinical study was conducted in Milan, and this is the response of the patients to the combined treatment, trastuzumab and pertuzumab, and lapatinib, sorry. And you can see that actually many patients responded with the tumor shrinkage. And these were like at least 4 or 5 lines of therapy, metastatic patients in which HER2 was found amplified. They actually had no therapeutical options and an expected life of very few weeks or months. And actually some of them, you can see one of them remained disease-free for years, actually. So this proves how you can use the models to understand how to properly treat a particular genetic alterations. And this, of course, now is ongoing on different models, on different alterations, and of course not only in colorectal cancer. Another example is, what if we do RNA sequencing of these models of colorectal cancer? So in the lab and in the institute, we have been generating a lot of RNA expression profiles to study the function, okay, from tumors, from more than 500 xenografts, from more than 200 cell lines, and from more than 100 organoids. Okay, so again, we have a big cohorts in which we can find specific alterations, specific features, and see what happens. And so this can be used for different things. But there is one advantage of a tumor grown in the mouse that is very important. So when you profile the RNA from this tumor, and the same is true for DNA, the RNA from cancer cells is human, because the tumor is a human tumor. But the RNA from stromal cells actually is from the mouse, because fibroblast, endothelial cells, immune cells are all from the mouse. So you can discriminate by the sequence, which is the transcriptome of the cancer cells and which is the transcriptome of the stromal cells. Like if you were able to dissect and profile separately. So you can generate a single mixture, an ASEC, and then by mapping, and this is bioinformatics, you separate human and mouse, so you see only cancer cells and only stromal cells. And you can do this for a number of things. You can be more precise in using transcriptomics to classify cancer cells. You can find cancer cell-specific targets, or you can just focus on the stroma and classify the different types of stroma in different tumors, which is becoming more and more important, and find targets in the stroma. Or finally, you can study tumor-stroma interactions. No? So, as an example, with the stroma, we were able to use this transcriptome to generate signatures that enable, when you profile a human tumor, to understand what stromal cells are abundant. I'm just skipping this. This is a complex way that this is how we do it. But then finally, you get signatures, like a signature for cancer-associated fibroblast. And tumors that has this fibroblast have this particular biology, a lot of active reactive stroma and few cancer cells. Endothelial cell score is a highly vascularized tumor. You can see the red blood cells around here, highly vascularized. Or leukocyte score is a tumor which is infiltrated in leukocyte. And you can get that from just a snapshot of RNA-seq of the tumor. The tumors that have all three scores low are just have a very tiny stroma. But these tumors, like triple high score, you see just few nests of cells in a very heterogeneous stroma, and this is not an artifact of sample. This is the biology of that tumor. These tumors tend to be the worst. Another example is to focus on cancer cells. So what if we use the cancer cell transcriptome to classify colorectal cancer? So we managed to do that, and we found a classification which is subdivides colorectal cancer in 5 subtypes, colorectal cancer intrinsic subtypes by their expression profiles. And you can see when you analyze matched xenograft and tumor of origin, they tend to remain of the same class, you can see. So, unfortunately, yes, most of them. And in particular, I would like to focus on one class, which is CRISB, which has poor prognosis. So these tumors are aggressive, but they are aggressive not because of the stroma, but because of the cancer cells. Another classification, which is called CMS, colorectal molecular subtypes, actually you can see subdivides the tumors by their microenvironment. So this is rich in leukocytes, this is poor in stroma, as this, and this is rich in stroma. So this is a classification which is not actually classifying cancer cell, it's classifying the stroma of the tumor. While CRIS, you can see it has some high and low stromal tumors in all groups. What are the diagnostic implications? Well, the diagnostic implication is that if you take a tumor and you sample different regions, and this was also published in the Journal of Pathology, you can see that with the CRIS subtype, it's always CRIS A. Always CRIS A, independently of where you sample. You can have areas with different amount of stroma, and you can see, while another classification, more classical, changes the class, depending on where you sample the tumor. So, of course, if you want to use it clinically, you need to be able to rely on 1 or maybe 2 samples, but not many samples. And CRIS B is associated with poor prognosis, you can see it here. And it is independent of the stage. This is Duke's classification, but it works in other types of classification, but also independently of the treatment. So these are low-risk cases that have not been treated. These are high-risk cases which have been treated with adjuvant treatment, and still CRIS B is worse, which means it's not just more aggressive, it's also more resistant to treatment. So this subtype is a subtype for which we have to find alternative treatments. And so this is, I think I made the examples that I wanted to make. Now I want to Sorry, there is a problem here. Yes. I want to just spend the last 5 minutes in describing what's happening at the European level in the field of cancer and cancer precision medicine and research. So a big project has been launched a year ago, which is called CanServe. It is an infrastructure project, so many different research infrastructure join forces to provide services to the community focused on cancer. And the services involve different type of support that you can obtain free because it's paid for by the EU. Disease models, advanced technology platforms, and you will see, so access to advanced technological platforms, research and development for biomarkers, research and development for new therapeutic solution, support in the design of complex clinical trials, and then this work package, which is acceleration translation into precision oncology. So you can imagine these services supporting a number of findings, and then we need to bring these findings to the patients. And this is actually what I am coordinating, this work package. So in particular, sorry, now I understand how it works. Let me make an example. So in the case of cancer models, of course, we participate as an infrastructure, which is called EuroPDX. So you can access models, like PDX models, but you can access xenografts in zebrafish, or you can ask to generate organoids, to perform genome editing, to obtain patient-derived cell lines, and other things. There is a lot of services. It's a big catalog, okay? And so you can obtain models either pre-existing or ask for models to be developed of your interest. Advanced technology, well, we can provide in vivo drug efficacy studies for problems of your interest. Imaging, a lot of advanced imaging facilities are made available. Genomic profiling, multimodal molecular imaging, micro-PET CT. This is not available in many centers, but you could actually, if you want to obtain imaging data from your models, you can ask for this service. Biomarker research, again, is a big field where maybe you have some idea, you have some preliminary data, you need to validate them, you need to consolidate them, and you ask for support on this huge list of services that you can see here. New therapeutic solutions, well, actually, there is a lot of immunotherapy support, you can see here, CAR-T, CAR-NK, drug screenings that can be done on models of interest. So it's a lot of effort here to support research in the validation of new drugs. But what is the field now? You're not a company, you do not have a new drug under development. But you may want to repurpose, like we did for colon, a drug which is used in breast cancer, you want to validate is used for colorectal, so you need support for that. And this can provide support. And for accelerated translation into personalized oncology clinical practice, I would like to briefly speak about a European molecular tumor board network that we have been assembling over the last few months. So typically, the entity that decides how to treat a patient based on genomic data is the molecular tumor board. But the way molecular tumor boards operate are not standard. The results are not traced, so we cannot really understand if how much things are working or not for the patients. So we decided to build up a network in which all the new findings that are provided through the infrastructure, plus all the new findings that are published, converge in a standardized way to the knowledge of the molecular tumor boards so that they can make decisions based on evidence, on existing evidence, and in a standardized way. We can also support as a service with the knowledge about putting together a new molecular tumor board in an institute that is interested. We can provide regulatory support, access to drugs and use of the drug is not always easy, and innovation support. So discoveries that are made, there is an infrastructure which is dedicated to provide you support to understand whether it is feasible and worth protecting your innovation. So To achieve, the services are up now. There is a call open. It closes the 4th of January. You need to go to this website, cancerb.eu, and browse and find the catalog. It is very simple. We also just launched a webinar last week, so you can actually understand how to apply. The users submit the application. The applications are evaluated and selected, and then the services are provided free of charge. And this is particularly important for young investigators who are just ramping up their labs and need support. Of course, you can write me if you want advice. This is my email. It's already at the beginning of the talk. And I think that's all. Okay, this is my group for some of the things that I've showed you. We are doing other things now, including immunotherapy. But I thought it was important to give a general overview of where the field is moving with all the problems related to precision oncology in cancer. Thank you very much.